Hello everyone, and thank you for the opportunity to present my work at this conference. Today I will talk about transcription factor co-binding partners and their identification with non-negative matrix factorization. In a cell, gene expression is regulated through various mechanisms, including binding of transcription factors. Binding of the individual transcription factors is quite well studied. However, usually transcription factors do not act alone, they cooperate instead. One of the most known cooperation events is co-binding of SOX2 and OCT4 transcription factors. They bind their consensus motif and promote pluripotency genes. However, it has been shown that OCT4 can switch partners and co-bind with SOX17 instead. Interestingly, in such case, they prefer binding a compressed motif, which is shorter from the canonical one by one spacer in between the two motifs. In addition, OCT4, in combination with SOX17, is involved in activation of primitive endoderm genes. So it is important to understand how different transcription factors co-bind DNA. One of the ways to do that is to investigate DNA binding motifs, motif orientations, and spacing. There are already some tools to discover transcription factor binding motifs. Some of them are general motif discovery tools that are good for individual motif de novo discovery, but unfortunately they cannot take into account another transcription factor binding motif, and they do not automatically summarize the possible cooperativity. On the other hand, there are some tools specifically intended to analyze TF cooperativity, and such tools search for co-binding pairs or co-binding motifs with different spacings and orientations. However, these tools might require more than one dataset as input or a known motif library in order to discover these co-binding motifs. Originally, the idea of using non-negative matrix factorization together with one hot encoded sequences was conceived in Boris Lenhardt's lab, where Sarvash Nikum developed a tool Archer that is able to uncover promoter architecture. We use the same idea in our approach, co-bind, where we discover co-binding motifs relative to the anchor transcription factor binding sites, and we summarize their spacings and orientations. The pipeline requires only one bed file. It can take more than that, and in that bed file, a user should have anchor transcription factor binding sites in bed format. The goal here is to look for co-binding motifs with fixed spacers upstream and downstream relative to these input anchors. In order to do that, we generate upstream and downstream flanking sequences and one hot encode them. Then, so our goal here is to kind of find these patterns in the sequences that could mean transcription factor binding sites, and these would be those co-binding motifs that we're looking for. So we use these sequences as input for the NMF algorithm, and here our input sequences is our data, where a non-negative matrix factorization then can factorize the matrix into two amplitude and pattern matrix, and dependent on how many components we ask, in this case it's 5, we can build candidate cobinder motifs. So we get 5 candidate cobinder motifs, and as you can see here, 2 of them really look interesting if we are talking about transcription factor binding, and the rest are not so interesting. So our next step after a non-negative matrix factorization is motif filtering. We use Gini coefficient to select only the most interesting motifs, and also motifs are trimmed to get rid of the zero IC positions or low IC positions. The key moment in the pipeline is also the motif clustering. So after NMF, since NMF is performed on a range of components, we get a redundant collection of candidate cobinder motifs. So we use motif clustering in order to reduce that redundancy and extract unique motifs. And then finally, we can summarize over one data set or a group of data sets, whatever the user prefers. So in this last step, as you can see here in this summary plot, we have our input anchor transcription factor binding motif that we had here as input, and motifs of cobinders. And each square here indicates where the cobinder can be found, either upstream or downstream, and the color intensity and where the square is will indicate the spacer, how many spaces in between the two motifs we have, and color intensity will say how many data sets have this particular configuration of anchor and cobinder motifs. And also we have a fraction of sequences with that specific combination configuration. Also, the pipeline will give the user those specific regions with the cobinders. So these can be used in downstream analysis. The data that has been used in our analysis is the newest version of Unibind data, which includes high confidence direct transcription factor binding sites from 7 species, and we have for more than 400 unique transcription factors. For our downstream analysis, so after we discovered these combining motifs, we used physical protein-protein interaction data from string database and Jasper and SysBP motif collections. So as a first example, I would like you to show what we get after we use SOX2 as our anchor motif. So here, in different species, we discover that SOX2 has one cobinder motif, which is upstream with a 0 spacer, so immediately after our anchor. But when we look into different species, we see that the same motif actually can be found in mouse and human data with the same spacing information. So in SOX2 case, it is quite obvious that the cobinder should be OCT4 or pool 5F1. However, using Jasper, SysBP, and string data, we can actually validate this combining event. So we are using Jasper and SysBP motif collections to compute motif similarity between our candidate cobinder. And here we want a very significant motif, which would give us the highest minus log 10 p-value. Here, and from string database, we use a combined protein-protein interaction score. So the higher the score is, the more likely is the interaction between the two proteins. So in this case, as you can see here, at the top right, in the top right corner, we actually have this interaction between SOX2 and OCT4. So we can validate this discovered cobinder. Another interesting thing that we can observe after we discover our cobinders is the difference between the conservation patterns. So here we have results from human, and in this plot on the left, you see SOX2 regions that do not have the cobinder, so our pipeline does not discover any cobinders. And here we have regions with discovered cobinder. So with this combining event. And as you can see here, we have an additional spike for the OCT4 motif, which is quite interesting. And in mouse, we can see a very, very similar pattern here. So as I mentioned briefly before, our pipeline is able to summarize the combining motifs that were discovered by the non-negative matrix factorization over a dataset or a group of datasets. So previously I was summarizing over all the SOX2 datasets, while here I'm summarizing the discovered combiners over all SOX related factors. So here the specific combinder that I have showed you before actually is found not only for SOX2 as an anchor in human and mouse, but also for SOX17 as an anchor in mouse. And if we look a bit closer in that into that specific case, we see that one of the spacers is specific for SOX2 when we use SOX2 as an anchor, and another spacer is when we use SOX17 as an anchor. And if you remember from the beginning of my talk, we know that OCT4, so our discovered cobinder, is able to switch partnering from SOX2 to SOX17 and activate a different set of genes. So, and we know that these two combinations of transcription factors are binding different motifs. And here I just want to show that we actually get these two combinations of motifs for these two combinations of transcription factors. So we have a canonical motif for SOX2 and OCT4, and then we have a compressed motif for SOX17 and OCT4. So we can recapitulate the results from the literature. Another example I would like to show you today is the cobinders for CTCF. In this case, in different species, we have two different combining motifs discovered with the various spacers. And if we look into different species, as you can see here, in mouse and human, we have same cobinders, and then we have one of the cobinders in zebrafish. But more interestingly here, we notice that the in human, we can find these cobinders more often, almost in all the data sets that we use as input. And again, with CTCF, the conservation patterns are also very interesting. So here we have only human results. So for the first cobinder, as you can see here, even we have a spike for the cobinder compared with no cobinder, and then even the CTCF regions where CTCF motif is found is more conserved compared where there is no cobinder. And with the second cobinder, the differences are not maybe that striking, but as you can see here, for another cobinder, we also have a small spike here. And the results in mouse look very similar. So overall, Combined is able to uncover non-combining motifs, but also quite a lot of potentially new cooperation events that cannot be validated through motif similarity and protein-protein interactions. So here, these non-validated events are in light pink. One of the examples of such event could be ASCL1 cobinder. So we have our anchor motif here and the possible cobinder here. So if we look at this motif, it actually looks quite similar to the FOXA1. And then when we do the motif similarity analysis, we get FOXA1 as the most similar motif, as the most significant match here. In a recently published paper, it has been shown that ASCL1 and FOXA1 are occupying the same regions in the genome. So we hope to look into these potentially new combining events and further analyze them and maybe to discover some new biology. In summary, Combined is discovering combined their motifs relative to the anchor, even in the single data set, and the only input required for the motif discovery is anchor transcription factor binding sites. After analyzing thousands of data sets, we demonstrate known and potentially new combining events. Some of these events are recurrent across species and recurrent within transcription factor families. Furthermore, we observe different conservation patterns in regions with combiners compared with regions without one. And finally, I would like to thank my lab for the support. Especially, I would like to thank my supervisor, Anthony Metalier, and my colleagues, Jaime and Tim, who are working with me on this project. Combined is currently under development, and if you'd like to know more about it, you can also visit my poster that I have in this conference, or just ask me questions.